Harvard's secret: how to reverse biological age by 3 years in 8 weeks. In 2022, a research team at Harvard Medical School published a paper that made headlines in scientific journals around the world, and almost none in mainstream media. They took mice and artificially accelerated their biological aging by disrupting their epigenome, the system of chemical instructions that tells your DNA which genes to activate and which to silence. The mice aged rapidly. Their eyes deteriorated, their muscles weakened, their cognitive function declined. They looked and functioned like old animals. Then the researchers reversed the process. They used a specific set of genes, the Yamanaka factors, which won a Nobel Prize in 2006, to partially reset the epigenome. The mice got younger, not metaphorically, measurably younger. Their vision improved, their muscle function improved, their brain function improved. Their biological age, as measured by established molecular clocks, decreased. The lead researcher on that paper was Dr. David Sinclair, the same Harvard scientist whose work on NAD+ and sirtuins we discussed in our episode on cellular energy. And the central conclusion of that research is one of the most profound statements in modern biology. Aging, at the cellular level, is not a one-way street. It is a loss of epigenetic information, and that information can, at least partially, be restored. Now, I want to be absolutely clear. We are not at the point where humans can reverse their biological age the way those mice did. But what is available to us right now, through specific, measurable, science-backed lifestyle and nutritional strategies, is the ability to slow and, in some cases, meaningfully reverse the rate at which our cells are aging. And we can now measure it. We can put a number on it. Today, I'm going to bring this entire series full circle. We are going to talk about what biological age actually is, how it is measured, what drives the gap between your biological age and your chronological age, and what the research identifies as the most powerful levers for keeping your cells younger than your birthday says they should be. We will dive into all of that in today's episode. I am Dr. Alexander Wellrich. Over the past several episodes, we have gone deep into the specific systems that govern how you age: your mitochondria, your gut microbiome, your inflammatory status, your NAD+ levels, your muscle mass, and your sleep architecture. Today, I want to give you the synthesis, the framework that ties all of those threads together into a coherent picture of what biological aging actually is and what you can do about it. Because here is what I have observed in 30 years of clinical practice. Chronological age is a poor predictor of how people actually feel, function, and look. I have had patients in their early 60s who, by every physiological and molecular measure available, were functioning like healthy 45-year-olds. And I have had patients in their early 50s who were biologically closer to 70. The difference was not luck or genetics alone. It was the sum of choices made at the cellular level, consistently, over time. The science of biological aging has advanced more in the last decade than in the previous century. We now have validated tools to measure it. We have mechanistic understanding of what drives it, and we have growing evidence of what slows it. That is what today is about. If this series has been valuable to you, if any of these conversations have given you something you could actually use, please subscribe and leave a comment. Your engagement is what allows this channel to reach more people who need this information. And now, let us talk about the age that actually matters. Let me start with the foundational question. What does it actually mean to have a biological age that is different from your chronological age? Your chronological age is simply the number of years since you were born. It is fixed, linear, and the same for everyone born on the same day. Your biological age is something entirely different. It is a measure of how your cells are actually functioning, how much damage has accumulated, how efficiently your repair systems are operating, how well your cellular machinery is maintaining itself. Two people born on the same day can have biological ages that differ by 10, 15, or even 20 years. Until recently, biological age was estimated through indirect proxies: grip strength, cardiovascular fitness, cognitive testing, blood biomarkers. These are useful but imprecise. The revolution in the field came with the development of epigenetic clocks. Your epigenome is the system of chemical modifications to your DNA, primarily methyl groups attached to specific positions on the DNA strand that control gene expression. These modifications change in predictable patterns as you age. In 2013, a biostatistician at UCLA named Steve Horvath discovered that by measuring the methylation pattern at 353 specific sites across the genome, he could predict a person's chronological age with remarkable accuracy, and the deviations from the expected pattern revealed biological age. If your methylation pattern looked 5 years older than expected for someone your chronological age, your biological age was 5 years older. If it looked 8 years younger, you were biologically 8 years younger. Horvath's original clock, now called the Horvath clock, has been refined through multiple generations. The second-generation clocks, including the pheno-age clock developed by Dr. Morgan Levine at Yale and the Grimm age clock developed at UCLA, incorporate additional biomarkers and more accurately predict health outcomes, not just chronological age, but risk of specific diseases and mortality. A study published in Aging Cell found that biological age, as measured by these epigenetic clocks, was a significantly stronger predictor of mortality, chronic disease risk, and functional decline than chronological age alone. Your birthday is a rough estimate. Your methylation clock is a molecular readout of what is actually happening inside your cells. These tests are now commercially available. Companies including True Diagnostic, Elysium Health, and others offer epigenetic age testing from a blood or saliva sample. Prices have come down significantly in the last few years. I am not endorsing any specific company, but I do believe that having a baseline biological age measurement and then retesting after a period of intentional intervention is one of the most motivating and informative things a health-conscious adult over 50 can do. Now that you understand what biological age is and how it is measured, let me walk you through the primary drivers of biological age acceleration, the factors that cause your epigenetic clock to run faster than it should. The first and most powerful driver is chronic inflammation, the inflammaging we discussed in our third episode. A study published in Nature Aging found that inflammatory markers, particularly interleukin-6 and C-reactive protein, were among the strongest predictors of epigenetic age acceleration across a large study population. Chronic inflammation does not just damage tissues. It directly alters epigenetic methylation patterns in ways that accelerate biological aging at the molecular level. Every percentage point reduction in your inflammatory markers is a step toward a younger biological age. The second driver is mitochondrial dysfunction. Your mitochondria do not just produce energy. They are deeply intertwined with the epigenetic machinery that maintains cellular identity and function. Research published in Cell Metabolism found a direct bidirectional relationship between mitochondrial health and epigenetic aging. Cells with dysfunctional mitochondria show accelerated epigenetic aging. Cells with healthy, abundant mitochondria maintain younger epigenetic profiles. Everything we discussed in our first episode about mitochondrial health connects directly to biological age. The third driver is chronic psychological stress. A study published in the Proceedings of the National Academy of Sciences found that adults who had experienced chronic psychological stress, measured through perceived stress scales and cortisol markers, had epigenetic ages that were significantly older than would be predicted from their chronological age. Chronic stress does not just feel aging, it measurably accelerates the epigenetic clock. Conversely, stress reduction practices, mindfulness, social connection, time in nature, purposeful engagement, have shown in preliminary research to slow or partially reverse epigenetic aging. The fourth driver is poor sleep quality, specifically the reduction in slow-wave sleep we discussed in our last episode. A study published in Aging found that sleep duration and quality were independently associated with epigenetic age acceleration. Adults sleeping fewer than 6 hours showed faster epigenetic aging compared to those sleeping 7 to 8 hours, even after controlling for other lifestyle factors. The glymphatic clearing that happens during deep sleep and the hormonal recalibration that occurs across a full sleep cycle are not just good for how you feel in the morning. They are protecting your epigenetic integrity night by night. The fifth driver is excess body fat, particularly visceral fat. Adipose tissue, especially the metabolically active fat stored around the abdominal organs, produces a constant stream of pro-inflammatory cytokines and disrupts hormonal signaling in ways that directly accelerate epigenetic aging. A study published in Genome Biology found that obesity was associated with epigenetic age acceleration of approximately 3 years on average, with the effect concentrated in the metabolically disruptive visceral fat compartment. And the sixth driver, which connects to everything we have discussed across this series, is sedentary behavior. Not just the absence of exercise, the presence of prolonged sitting and physical inactivity as a consistent daily pattern. A large study published in the American Journal of Epidemiology found that adults with high levels of sedentary time had measurably accelerated epigenetic aging, independent of whether they exercised at other times of day. Movement throughout the day, not just structured exercise sessions, matters for biological age. A few years ago, I had the opportunity to work with a patient I will call Patricia. She was 62, a retired pharmacist from San Antonio, Texas. What made her case particularly illuminating was that she came in with her identical twin sister Helen, who lived in the same city and had grown up in the same household. Patricia and Helen shared essentially identical genetics, same birthday, same childhood environment, same genetic starting point. But by 62, they looked and functioned differently enough that people often guessed Patricia was younger than her sister by 5 to 8 years. Their epigenetic age tests confirmed what was clinically observable. Patricia's biological age tested at 54. Helen's tested at 68. A 14-year gap in identical twins, driven entirely by the choices each had made over the preceding 3 decades. Patricia exercised consistently, ate a Mediterranean-style diet rich in plants and fish, had never smoked, slept 7 to 8 hours per night, had maintained a close social network, and had practiced what she described as deliberate stress management since her 40s. Helen had led a more sedentary life, eaten a more processed diet, had decades of disrupted sleep as a night shift nurse, carried significant visceral weight, and had experienced years of chronic occupational stress without recovery practices. Same genes, 14 years of biological age difference. The environment won, comprehensively. But here's where the story gets genuinely hopeful. Because the research is now showing that biological age is not just a measurement. It is a target. And it moves in response to the right interventions, sometimes faster than researchers expected. This is the part of the conversation I find most exciting as a clinician, because we are now moving beyond correlation into intervention evidence. Studies that have actually measured biological age before and after specific lifestyle changes and documented the shift. The most compelling intervention study to date was published in 2021 in the journal Aging by Dr. Fitzgerald and colleagues. They conducted a randomized controlled trial in which middle-aged men were placed on an 8-week program combining a specific dietary protocol, high in leafy greens, cruciferous vegetables, colorful produce, and liver-supportive compounds including methylation nutrients, with targeted exercise, sleep optimization, and stress reduction. At the end of 8 weeks, the intervention group showed an average reduction in biological age of 3.23 years compared to controls. 3 years of biological age reduced in 8 weeks through lifestyle intervention? That is not a small signal. The dietary components with the strongest evidence for epigenetic age reduction include methylation-supporting nutrients, specifically folate, found in leafy greens and legumes, choline, found in eggs and liver, and betaine, found in beets and spinach. These nutrients support the methylation chemistry that epigenetic clocks measure. When the building blocks of healthy methylation are abundant, the epigenetic machinery maintains itself more accurately. Polyphenols, the plant compounds found in berries, green tea, extra virgin olive oil, dark chocolate, and turmeric, have been shown in multiple studies to influence DNA methylation patterns in ways that are associated with younger biological age. A study published in Nutrients found that higher dietary polyphenol intake was independently associated with younger epigenetic age in a large cohort of adults over 50. These compounds are not just anti-inflammatory, they are directly influencing the epigenetic machinery. Exercise, and specifically the combination of resistance training and aerobic activity we have discussed throughout this series, has been shown in a growing body of research to reduce epigenetic age. A study published in Aging Cell found that master athletes in their 60s and 70s had epigenetic ages that were significantly younger than sedentary peers, and the gap was not explained by genetics, but by sustained exercise history. A separate study from King's College London found that adults who exercised regularly had biological ages up to 10 years younger than sedentary peers of the same chronological age. Time-restricted eating, specifically a consistent 10 to 12-hour eating window, has shown preliminary evidence of epigenetic age reduction. The circadian alignment of eating, the activation of autophagy during the fasting window, and the reduction in oxidative stress from constant caloric processing all converge on mechanisms that protect epigenetic integrity. And social connection, which I want to address directly because it often gets treated as a soft lifestyle factor rather than a biological one. A study published in the Proceedings of the National Academy of Sciences found that social isolation was associated with epigenetic age acceleration, comparable in magnitude to smoking. The reverse was also true. Adults with strong, purposeful social networks showed slower epigenetic aging. Loneliness is not just psychologically damaging, it is molecularly aging. Let me give you an honest picture of where supplements fit in the biological aging framework, because the evidence here is more nuanced than marketing would suggest. NAD+ precursors, NR and NMN, which we discussed in depth in our 4th episode, have mechanistic support for slowing epigenetic aging through sirtuin activation. Sirtuins are directly involved in maintaining epigenetic integrity. A study published in Cell demonstrated that declining NAD+ levels in aging mice were associated with epigenetic dysregulation, and that restoring NAD+ partially corrected those patterns. Human trials specifically measuring epigenetic age as an outcome are still limited, but the mechanistic chain from NAD+ to sirtuin activity to epigenetic maintenance is well established. Alpha-ketoglutarate, AKG, is a compound involved in cellular energy metabolism that has received recent attention for its effects on biological age. A study published in Cell Metabolism found that AKG supplementation extended health span in mice and produced measurable reductions in epigenetic age markers. A human trial published in Aging found that a calcium alpha-ketoglutarate supplement produced an average reduction of 8 years in biological age as measured by the Horvath clock in a cohort of adults between 42 and 78. This is a single study and requires replication, but it is being taken seriously in the research community. Omega-3 fatty acids have epigenetic evidence specifically. A randomized controlled trial published in Brain, Behavior, and Immunity found that omega-3 supplementation produced changes in DNA methylation patterns associated with reduced biological aging, alongside reductions in inflammatory markers. For a supplement with an already outstanding safety profile and broad cardiovascular and anti-inflammatory evidence, the addition of epigenetic support makes the case for omega-3s even more compelling. Vitamin D is worth mentioning in this context because it functions more like a hormone than a traditional vitamin, regulating gene expression across thousands of genes in virtually every tissue in the body. Vitamin D deficiency has been associated in multiple studies with accelerated epigenetic aging, and supplementation in deficient individuals has shown preliminary evidence of epigenetic correction. I want to be clear. Supplements are the top layer of the pyramid, not the foundation. The foundation, the diet, the exercise, the sleep, the stress management, the social connection, does more for biological age than any supplement stack. But for people who have built that foundation, these compounds represent a reasonable, evidence-informed addition. I want to close the core content with something that I believe belongs in every serious conversation about biological aging. And that is conspicuously absent from most of them: purpose. Research published in JAMA Psychiatry found that adults who reported a high sense of purpose in life had measurably slower epigenetic aging compared to those reporting low purpose, independent of diet, exercise, income, or social network size. A separate study published in Psychological Science found that purpose in life was associated with longer telomeres, the protective caps on chromosomes that shorten with biological aging, and with preserved mitochondrial function. We discussed in our gut health episode that Columbia University researchers found a direct link between positive psychosocial states and mitochondrial enzyme activity. Now we are seeing convergent evidence from epigenetic research that purpose, meaning, and positive engagement with life are not just philosophically important, they are biologically protective. They slow the molecular clock. Dr. Viktor Frankl, the Austrian psychiatrist who survived the Nazi concentration camps and founded logotherapy, wrote that the primary human drive is not pleasure, but meaning. The biology is now beginning to confirm this at the molecular level. Humans who live with a sense of purpose, who feel that their actions matter, that they are connected to something larger than their immediate circumstances, maintain healthier epigenomes and longer telomeres than those who do not. For adults over 50, this is not an abstract philosophical observation. It is a direct prescription. Volunteer work, mentorship, creative engagement, caregiving, spiritual practice, community involvement, whatever form purpose takes for you, these activities are not peripheral to your health. They are part of the biological protocol. What gives you a reason to get up in the morning? That question is not soft. It is one of the most medically important questions I can ask a patient. Here is your integrated action plan, the synthesis of everything we have covered in this series, framed through the lens of biological age. 1. Build your plate around methylation-supporting nutrients every day. Dark leafy greens, spinach, kale, arugula, at least once daily. Eggs 4 to 5 times per week for choline. Beets, legumes, and cruciferous vegetables regularly. These are the raw materials for healthy epigenetic maintenance. 2. Commit to resistance training 2 to 3 times per week and add consistent low-intensity movement throughout every day. Take the stairs. Park farther away. Walk after dinner. The research shows that sedentary time accelerates epigenetic aging independent of exercise. Movement throughout the day, not just in gym sessions, matters molecularly. 3. Prioritize sleep with the full protocol from our last episode. Morning light, consistent timing, cool bedroom, evening light reduction, magnesium before bed. 7 to 8 hours is not a luxury. It is epigenetic maintenance. 4. Manage your inflammatory load actively. This means the food swaps from our inflammation episode: extra virgin olive oil, fatty fish, berries, leafy greens, and the lifestyle practices that reduce inflammatory signaling. Stress management, adequate sleep, reduction of ultra-processed foods and refined vegetable oils. 5. Invest deliberately in your social connections and sense of purpose. Schedule the phone calls, the dinners, the community involvement, the volunteering. These are not nice-to-haves. They are biological age interventions with measurable molecular effects. 6. Consider getting a baseline epigenetic age test. Companies like True Diagnostic, Elysium Health, and others offer these at increasingly accessible price points. Having a number, knowing where your biological age sits relative to your chronological age, creates a feedback loop that makes every other intervention more motivating and more measurable. 7. Have conversations with your physician about the biomarkers that matter most for biological aging. HSCRP for inflammation, 25-hydroxy-vitamin D, fasting insulin, NAD+ precursor status, and omega-3 index. These are not exotic tests. Many are covered by insurance. They give you a cellular dashboard and a baseline to improve from. Let me bring this series to its conclusion. We have spent 6 episodes going deep into the specific systems that govern how you age: your mitochondria, your gut microbiome, your inflammatory status, your NAD+ levels, your muscle mass, and your sleep architecture. Today we brought those threads together into the framework that contains all of them: biological age, the molecular readout of how your cells are actually functioning, and the target that all of those interventions are ultimately moving. The science is clear. Biological age is not fixed. It is not determined by your genetics alone. It moves in both directions, in response to what you eat, how you move, how you sleep, how you manage stress, who you are connected to, and what you believe your life is for. Patricia and Helen proved it. The Fitzgerald trial proved it. The exercise and epigenetic literature proves it. The Harvard mice got younger. We cannot yet do exactly what those researchers did, but we can do something that, in practical terms, may matter even more. We can make choices every day that move our biological clock in the direction we want it to go. You are not the age on your driver's license. You are the age your cells are behaving. And that number is something you can influence, starting today. If this series has given you something genuinely valuable, if any one of these episodes has changed the way you think about your health, or given you a tool you have used, please take a moment to like, subscribe, and share this channel with someone who needs it. Every comment you leave, every share, every subscription allows us to reach more people who deserve this information. It has been my honor to be your guide in this series. This is not the end. There is so much more to cover, and I look forward to continuing this conversation. Until next time, take care of yourself from the inside out.